let's do it. Thank you, everyone, for coming. This is our first ever Geoscience Grab Bag. So our panelists today are going to be speaking about a number of different topics. And so our speakers are Kimberly Foecke from George Washington University, Elie Gurarie from the University of Maryland, Patty Ryberg from Park University, Ingo Sonder from the University at Buffalo, and Jaemon van de Hoek, who is joining us remotely via Zoom from Oregon State University in Corvallis. So I'll turn it over to these guys, and then we'll open it up for questions.
I think I've got a good enough view. All right, yeah, so I'm going to talk about some discoveries that we made about caribou migrations in North America. And just to sort of cut a little bit to the chase, so we discovered is this very large continental scaled synchrony in the start timing of migrations of these caribou. Let's see if this works, which is driven, it turns out, by kind of oceanic climate forcings. But the arrival time of the caribou, which in the spring migration, they have to get there to lay their calves, actually depends not at all on the weather during that spring or winter, but entirely on what was happening the previous summer. And the explanation for that is insects, and we'll see exactly how. So just briefly about caribou. Caribou, they have a circumpolar distribution. They're extremely important. They're not very abundant in the Arctic. They're the main source of protein and very important for the cultures and religions of all people in the north. In Eurasia, they're mainly domesticated and called reindeer. They're an ecological keystone. They're important both for predators and also they shape very much what is happening in the tundra growth-wise. And many of these populations are falling. So some populations in northern Canada, for example, where there are 500,000 maybe in one herd 20 years ago are now down to maybe 10,000. So there's some very precipitous collapses in caribou populations. Climate change, of course, is happening much more rapidly in the north than elsewhere. Resource extraction is expanding in the north related to climate change. So there's a lot of important questions. So one of the main tools we use for studying animals in the wild is satellite collaring. Here's a picture of my colleague Kyle Jolie in western Alaska. This is a caribou migrating across the Koyukuk River and kind of relatively painlessly being pinned to the boat and satellite collared. I'll note that this is done often in collaboration with local indigenous communities. And we've aggregated data sets from these 7 herds across North America. So this is kind of an unprecedentedly large movement data set extending 20 years in some cases, over 1,000 animals. And we were able to analyze these migrations. So this will show you an animation. In the upper figure we have 2013, in the lower panel we have 2014. And I don't know how to make this start. Let's try this. There we go. So here they are sort of in their winter ranges in the south across the bottom. And you notice here we're in April, and then all of a sudden they start taking off. And you can see that in 2014 how they took off much earlier than in 2013 in what it appears to be a large-scale synchronous way. And that was rather unexpected. There's no particular reason that we would think that they would all move together. And it turns out that a lot of the variability in that migration timing can be predicted by these large-scale oceanic climate forcings, like the Pacific Decadal Oscillation, which is sort of a measure of how much warm water is sloshing up in the Gulf of Alaska, or the North Atlantic Oscillation, which is a measure of where the high-pressure system is, whether it's over the North Pole or further south over North Atlantic. But, so in this figure, what we have here is the start date of the migration. So these are sort of late migrators that migrate, say, on May 15th. Some animals or some years, they would migrate much earlier. There's a lot of variability across years. But if they start late, they just move a lot faster. So for example, these late starters might migrate in 20 days to get their 300 miles, 400 miles sometimes to their calving grounds, whereas early starters might kind of dawdle, take a couple of months to get there. And that's because they really, really need to get to their calving grounds to drop their calves. And as it turns out, none of those climate predictors have any predictive value for their arrival time, but if it was a cool and windy summer, they would have early migrations. And our main hypothesis for this is insect harassment. Insects in the summer are the main drivers of caribou movements and behaviors. They spend most of their energy trying to avoid insects. So a cool and windy summer has fewer insects, healthier animals, an earlier fall rut. I put this in red because this is one link that's not very strong, but it definitely leads to an earlier migration arrival time and presumably higher calving rate in the following year. So in the far north, it's sort of, I say traditional, it's basically common knowledge among the indigenous communities up there that if it's a crappy summer for people, then the caribou are loving it. You know, if it's wet and cold and windy, then it's a good year for the caribou, mainly because there's a lot fewer bugs. And our main sort of result there was that good summers lead to earlier calving in the following year. So with that, I thank you for your attention.
I think it's my turn.
All right, so just
We'll just shift on to the next one. Right? Hey, whoa, went too far. Went too far. All right. My research deals with fossil environments down in Antarctica. For most of the history of the planet, Antarctica has actually been free of ice. So we're actually going down there to find ecosystems during a time when polar latitudes actually were covered with lots and lots of plant life. So the reason why we focus on specifically the Permian and the Triassic boundary with the plant fossils is because the largest extinction event in the history of our planet actually happened, I knew I was going to do this, actually happened at the Permian time period. Up to 90% of all life on the planet actually disappeared during this. I mean, here is the dinosaurs, and here's the end Permian. So there's a huge transition between what the world looked like in the Permian and what it looked like in the Triassic. So we're trying to figure out what sort of environmental changes do you see in high latitudes when you're going across a mass extinction. And another interesting thing to note is in the Permian and Triassic, we were actually going from an ice house to a greenhouse. And this is the last time in the history of the planet that we've actually gone from an ice house to a greenhouse, which is what we may be going through today. If you look at the graph down here, you can see we're still in an ice house, but as the Earth gets warmer and warmer, we could be transitioning to a greenhouse. So we're looking at this last time period in Earth's history when we had this transition from a cold climate into a warm climate to see sort of how our high latitudes responding to these changes. So we head to Antarctica because Antarctica has always been in high latitudes throughout geologic history, and there's not a lot of land coverage that we have to worry about digging through to get down to the fossils. So we don't have to worry about soil and trees and plants to get to the fossils. They're just sitting right on the surface. So while Antarctica really looks like this today, 250 million years ago at this extinction event, it probably looks something more like this. We've actually found fossil forests that with trees over 100 feet tall, and they were growing in thick stands in high densities during this time period. So we travel down to the Transantarctic Mountains and go camping so that we can collect these fossils to sort of reconstruct the environment 250 million years ago. Now, the reason why this is sort of difficult to do is because most of the plants that were alive at this time have absolutely no modern relatives today. I can't say, oh, the plants look like a maple tree, because there's absolutely nothing like them on the planet today. They actually belong to this group called the seed ferns that disappeared sometime before the dinosaurs actually really took over the planet, sometime in the Jurassic. So I can't say, oh, it'd be just like this ecosystem, because there is no real ecosystem like it today. But what we have discovered is that we have plants that are indicative of some sort of modern day environment. So a couple of the plants do have some modern relatives. They're very minor components of the ecosystem, but they do help but provide some sort of evidence of what the ecosystem was. Now, the biggest thing that everyone loves to talk about is there were actually forests in Antarctica 260 million years until about 30 million years ago. Antarctica was covered with forests. And we know this because the forests are still there today. This is an individual tree stump. I believe it's about 4 feet tall. And then in here, it's I've pointed out the different tree stumps that are still in the same spot they were in when they were actually growing. So they haven't been transported, they haven't moved, they're in the exact same spot as when they were alive. So doing this, we can actually figure out how dense these forests were. The actual cellular structure of these trees are still preserved. So we can do tree ring analysis on these things. And with the tree ring analysis, we've discovered that there's nothing like it on the planet today. These trees grew in a really weird manner. They grew and they grew and they grew, and then they shut down all of a sudden and went dormant. And trees don't do that today. So we're not sure. We've hypothesized that it wasn't the cold that shuts it down like it does today in northern latitudes, and it wasn't water availability because these things were growing in back swamps and water environments, but light is a true indicator because plants need light to survive, and for half the year there is no light in Antarctica. So this is sort of a unique ecosystem that doesn't exist today, but maybe will exist in the future as the climate continues to warm. Now, I said a lot of these plants, I can't point to any plants living today because there's nothing like them, which causes some problems for us because plants fall apart when they're still living. All the trees have lost their leaves, but are the trees dead? No, they're still alive. They'll come back and they'll do it, bring more leaves in. So we have parts of We have parts of plants that fall apart even though the plant hasn't died yet. So we have to come and figure out what pieces of each plant actually fit together. But we don't have a picture of the puzzle, so that makes things much more difficult. In our last trip down there, we actually found more leaves to show that this wasn't just one kind of plant down there. There's lots of these different types of plants down there. And we found their reproductive structures. We found the flowers or cones that were actually associated with these things. So we've managed to put two huge pieces of the puzzle together again. So most of what we know about these Antarctic fossil ecosystems is the canopy trees. We know what the trees were like, we know what the leaves were, we know how the leaves were attached to the tree, we know how the trees were growing, but we don't know what was growing underneath the trees. So one of the biggest puzzles we have to figure out now is what's going on underneath all of these trees. And in our last trip down there over last winter, we've actually found a lot of components of the understory of these ecosystems. We found lots of ferns in our Triassic after the recovery, and we haven't been able to find ferns in this locality that we went to previously. One of the really exciting and confounding things that we keep finding down there are cycads. Cycads are subtropical plants, and yet these things are growing at south polar latitudes when they were alive. So this is a huge indicator that Antarctica was warm enough for subtropical plants to be growing, even though it's still somewhere close to the South Pole. We found some other things as well, some non-seed plants that also help fill in the gap. We haven't filled in all the gaps because we have two completely different ecosystems before and after the extinction event, and so we have to try to reconcile two different ecosystems. And one of the neat things about Antarctic fossils is that they're actually anatomically preserved. So we actually have the cellular structure, the three-dimensional structure of these things still preserved in the rocks. So we can go through them cell layer by cell layer to help us reconstruct it instead of looking at a flat specimen that's been squished on a rock. So that's it's the rarest type of fossilization, and we've managed to find abundance of it in Antarctica. And then the biggest ecosystem issue that we've really talked about, I've mentioned, is how are these things living down there when the sun doesn't rise for half the year? So we have an environmental system that doesn't exist on the planet today, and we're trying to resolve how do plants and animals actually survive down there when the sun doesn't come up, and half the year it doesn't set either. So those are some of the puzzle pieces that we managed to put together, but we still have a lot more of the puzzle to go through and figure out. And I thank you for your attention. Oh, that's not for you. Thank you.
Okay.
Yes.
Now we're bringing in,
Jamo.
give us 1 second. I'm here if you can hear me.
We can. Can you hear us?
I can. It sounds great. I'm sharing my screen.
All right, give us 1 second to just get your screen up on the screen here. Okay, we can see your screen. Go ahead.
Thanks very much. I'll go full screen here. I can go full screen. Okay. Sorry I couldn't be there, but I really enjoyed listening to the first two talks so far. I'm going to be talking about progress that we've been making on a project over the last couple of years, which is basically taking a global survey of refugee camps and assessing the climatic marginality of these refugee camps, since that has, in some cases, some pretty new and perhaps cautionary tales about future climate change effects on vulnerable populations. So first of all, this is just a bit of background on the refugee situation. This is a global issue. As of mid-2018, there are nearly 20 million refugees under UN mandate, and that is the UN High Commission on Refugees, UNHCR. So these are camps, these are refugees that may live in camps, they may live in urban areas, but they're basically ostensibly under the protections of UNHCR. It's global, it's also a long-term challenge. Some of you may have heard this statistic about the average refugee camp has been inhabited by 17 years. That's a bit contentious, but probably not too far off base. By the UNHCR's own accounting, more than 2/3 of refugees live in what they call protracted refugee scenarios, in which basic rights and essential economic, social, and psychological needs remain unfulfilled after years in exile. So we have a global issue spanning dozens of countries. We have thousands of refugee camps, and we have these camps inhabited really generation after generation. These are the 5 countries, Turkey, Uganda, Pakistan, Lebanon, and Iran, that host the majority of the global refugee population. And we'll be talking about Turkey, Uganda, and Pakistan at the end of the presentation as sort of focused case study results. So camp siting and conditions really conform to local context. That is, in some cases, literally the topography, but more broadly, that's the social and political situation. We see a Rohingya refugee camp in Bangladesh. And as you can see, it looks drastically different from a Syrian refugee camp in Jordan. So both of these are designed, managed, and inhabited in a very local or at most national sort of context. So in no way do we have a global paradigm for refugee camps. It's highly variable. One thing that is consistent is refugee camps are commonly found in border regions. This graph here shows that over half of refugee camps are built within 50 kilometers of the border. That makes sense because refugees cross the border to seek asylum for protections from conflict. However, these camps end up being highly remote, geographically marginalized, and because of security concerns and other political constraints, many host countries warehouse refugees in remote camps and actually deny refugees the right of freedom of movement or wage-seeking employment. And if they don't outright deny it, they make it very difficult to gain the appropriate permissions to do so. So these restrictions not only limit economic opportunities, they obviously also affect livelihoods, poverty, likelihood. And in the case of our study, they require this means that refugee communities require special assistance to adapt to climate change effect. Now, what other populations have at their disposal is basically the right to leave. If things are looking rough, you leave. Many of us in the room probably are descended from migrants who made that same decision. And this is still happening every day all around the world. This is a resilience building decision to leave. In the case of climate change, we know that from recent research that higher temperatures and rainfall extremes correlate with increases in migration away from affected regions. But as we just mentioned, this is a problem in refugee camps where the inability to leave a camp, despite extreme exposure or vulnerability, means that that camp may effectively act as a climate trap. The climate scenario can be quite extreme, but the social and political conditions restrict movement away from that camp. And we see here 2 plots from a recent paper that show on the y-axis the likelihood of migration as we see, for example, increase in temperature on the left-hand side. So in our study, we took a global view of refugee camps. We looked at 922 refugee camps managed by UNHCR. Since camp siting and management is really decided, local and national level, we said, let's compare all these refugee camps to other sites within each camp hosting country and see basically if camps are sort of have marginal conditions in terms of these climate variables compared to other sites within a given country. So we conducted a stratified random sample to find these sites. And then we looked at 3 climate migration relevant variables at refugee camps at each site, both the camp and the random site. The first is annual rainfall, and here we see a map of the distribution of annual rainfall based on a product called CHIRPS. This is a screenshot from the platform we use to analyze everything, a cloud-based analytical platform called Google Earth Engine. Here's CMIP5 model output of future change in annual rainfall. And then we also see future change in near surface air temperature. So these 3 variables were selected because they come from the literature to know, we know that they have an effect on the willingness to migrate away. So let's look at some results. Globally, refugee camps have higher annual precipitation. We see the red distribution here relative to national level random sites. So we're throwing all of our countries together here. We also see that refugee camps have relatively higher modeled change in precipitation. Of note here, I don't know if you can see my pointer, but note that this distribution actually goes into the negative range. So a good share of camps, as well as our random sites, are actually expecting a loss of precipitation. In terms of surface temperature, then, we see roughly similar. So there's not necessarily too striking of differences, but when we look country to country, we start seeing really a high degree of variation in terms of how camps compare to other potentially viable sites within their country. If we look at Turkey, it's home to 4 million refugees, 3.5 million of which come from the Syrian civil war, which is about to enter its 8th year. By the way, this Turkey has only received 23% of its requested budget as of July this year. And in this case, we see that precipitation is likely to increase much higher in refugee camps on the left-hand side compared to random sites. We see that camps are also expected to see a much more extreme loss of precipitation and a little bit higher temperature change as well. So in all 3 of these, we see it's higher precipitation to begin with, but more loss of precipitation and higher temperatures. In Uganda, we're home to 1.2 million refugees, most of which are from South Sudan. Uganda only received 6% of its requested budget last year. And we see a completely different trend. In this case, precipitation and fractional precipitation change, and then temperature change, subtly different, but Uganda, we see, for example, a bit of a more positive picture, not nearly as extreme change at refugee camps compared to others. And in Pakistan, home to 1.4 million Afghan refugees, we see another, another example of how these climate variables can vary site to site. Here we see Pakistan also expecting refugee camps slightly lower precipitation change, but more extreme temperature change, right? So not all these variables always line up country to country, but when we look at individual camps, we can highlight individual camps that have extreme changes in precipitation and temperature. So effectively the highest ranking, the most loss of precipitation, or the most gain of precipitation, which could lead to flooding. We see the most temperature increase. These are the 3 sites, 3 refugee camps, country to country, that come out of this relative ranking. In Turkey, in Uganda, and Pakistan, these are the highest ranking. And you can see even here just how dramatically different these refugee camp spatial morphologies are. They just look completely different. And indeed, these are different from their neighbors. These are the most extreme, the sites that expect the most extreme change. So we've identified the most extreme change in precipitation and temperature. In the second bullet point, the big takeaway from this is that while refugee camps in some cases may not substantially differ from our comparison sites, they will perhaps face similar climate exposure, but they simply have fewer resources, including the right to leave the camp to address those challenges, thus reinforcing this climate trap scenario. So further development of the study over the next year, we're hoping that this will encourage the end of warehousing of refugees and provide viable pathways towards resettlement or integration. This also sets the stage for what hasn't been done by UNHCR, which is evaluating potential long-term vulnerabilities of refugee communities under climate change and the development of place-based solutions that simply won't fail given the context and local needs of refugees and the host countries. Thanks very much for your attention.
Oh, Kimberly? Foecke, Foecke, yes. Okay, so we're going to take a pretty big departure from what we just heard about here, and I'm going to be talking to you about the work that I'm doing for my doctoral dissertation in relation to using geochemical methods to reconstruct the diets of organisms in the past, specifically our human cousins who are now extinct, the Neanderthals. So isotope geochemistry is a set of techniques that are broadly useful to us in interpreting fossils. There are various different isotopes that can tell us different things about a fossil. You've probably all heard of radiocarbon dating, which uses the isotope carbon-14 to tell us something about the age of a fossil. There are many other chemical isotopes that are broadly useful to us. An example of this is strontium isotopes, which can tell us about migrations of whatever animal we're looking at in the fossil record, where they lived, where they grew up, where they moved around. And the one that I'm particularly interested in is the nitrogen isotopic system, which can broadly tell us something about diet. So diet is a really key driver in biological evolution, and it's a very key selective pressure for organisms. So being able to track diet accurately through the fossil record is really key to understanding the evolution of species, and in my case, the evolution of humans and the dietary practices that we have, and understanding why we're here and Neanderthals are not. So how does nitrogen tell us something about diet? Well, animals need nitrogen to survive. Almost every animal needs nitrogen in some form, but most animals cannot produce it within their own bodies. So animals need to get nitrogen by eating it in their food resources. So nitrogen starts out in the air, and bacteria take it up from the air, goes into plants, which are then consumed by herbivores, which are then consumed by carnivores. And what you see is nitrogen-15, the heavy isotope of nitrogen, being concentrated in higher ratios as you move up the food chain. So ultimately what we can see when we measure nitrogen-15 in the fossil record is a measure of roughly how much meat an animal was eating, whether it's a carnivore, an herbivore, or an omnivore, somewhere in between. So when we start applying this to Neanderthals, my work comes about because the isotopic evidence for Neanderthal diet doesn't agree with other measures that we have for Neanderthal diet. So when you look at Neanderthal sites, we see a really nice, neat stratification between herbivores with low nitrogen-15 values, omnivores somewhere in the middle, and carnivores like hyenas up at the very high end of nitrogen-15 concentration. When you plot Neanderthals on here, they actually exceed the values of carnivores that are subsisting entirely on meat diets. And this is a really strange result, and most researchers have taken this and interpreted this as Neanderthals are what we call hypercarnivores, meaning that they subsist almost exclusively on meat. They're eating nothing but meat. But when we look at other archaeological evidence, like plant fossils on the teeth of Neanderthals and remains that are in their fireplaces, the hearths that they cooked on, what we actually find is that Neanderthals plot closer to the omnivore level with bears. They're eating a pretty wide variety of plants in addition to their meat resources. We know they were eating meat. We have butchery. We have other things that tell us that. But the level of plants that they're eating is not trivial. So based on isotopes, Neanderthals are crazy levels of carnivore. They're eating just an unreasonably huge amount of meat. But based on everything else that we know, Neanderthals are more omnivorous, eating a diet more typical of a modern human today. We trust the other evidence, the plant evidence, that we have a little bit more than we trust the isotopes. It's pretty firm, and there's not really a lot to dispute the existence of plant fossils on Neanderthal teeth. So we wondered whether this means we have something wrong with how we're interpreting our isotope data in terms of Neanderthal diet. So this leads sort of to my hypotheses for this work. Neanderthals might not be just this picture of how they're getting their meat resources. Plants have really low nitrogen-15, so the high nitrogen-15 signal is coming from somewhere else, most likely meat resources. But in order to get the amount of meat that you see in the isotopic record, you would have to consume enough meat that it would probably kill you. So it's a little bit incongruous to assume that Neanderthals are just doing this, where they're hunting really large animals and subsisting on only that. So what if Neanderthals are also behaving sort of like a hyena and opportunistically scavenging carcasses that have been left out by other carnivores? Neanderthals are living in very marginal environments, similar to the high Arctic or tundra Siberia today. And if they happened upon a carcass that was even remotely edible, I can't imagine that it would be a good evolutionary strategy to pass up the opportunity to get that food resource. So we're hypothesizing that Neanderthals are getting their meat resources both fresh from hunting, but also possibly from scavenging. So how can we see this in the fossil record? Well, what we would expect to see is fresh meat having a baseline signature of some value and Neanderthals being offset from that, but meat that has gone slightly bad having a slightly different isotopic signal, presumably a little bit higher, which would bump the Neanderthal dietary signal up to where it might reflect something like what we end up seeing in the fossil record. So my research question is, does the decomposition process of this animal tissue cause the nitrogen-15 concentration to increase in the basic food resource? So my work is essentially experimental putrefaction, which is a fancy way of saying that I rot meat all day, every day. And this is preliminary work that I've been doing to measure nitrogen-15 throughout the process of decay of animal tissue and track change over time. So what I do is we place experimentally derived meat, essentially steaks that I get from a butcher who's been very accommodating and nice to me over the last year, in a rot box, which we have custom built and placed outdoors in a greenhouse that's climatically controlled. And we take samples from day 0 all the way through a couple of weeks of this process as this decay is occurring. And what we end up finding, this is a schematic because I can't actually show you any of my results because we have a publication pending, but we end up finding that during the initial rotting process, about the first week, where you've still got everything moist and there's a lot of microbial activity, nitrogen-15 concentrations increase at a statistically significant rate. As the first week is sort of winding down and you get a drying out process starting in about the second week of decay, this meat starts to look more like beef jerky, and there's a decrease in microbial activity, and it is reflected in a decrease in the nitrogen-15 concentration over time. So this is really interesting because it shows us that nitrogen-15 is mobile through these processes, which we didn't know before. And there's variation between the different processes of putrefaction, the different stages of putrefaction that are leading us to investigate further other things that might result in Neanderthals getting a high nitrogen-15 signal. So broadly, what does this mean for our understanding of Neanderthals and past diet? Well, first, it shows us that the nitrogen-15 dietary signal that has been applied really widely throughout the study of diet in the past by many, many researchers is much more complicated than we previously thought. We're also suspecting that Neanderthals may have been getting their food differently than all scientists prior to this had predicted. And that their food strategies might be a little bit more complicated than we give them credit for. And finally, we really need to keep working on understanding this isotope system. And I have many experiments in progress with cooking and rotting different sorts of tissues, and we're growing experimental plants to test some other aspects of this. This system is widely applied, and as we all know, science is measurement limited. And if we can't measure things properly and we don't know what our measurement reflects, we're not going to be able to answer questions very well. So if we can fine-tune the measurements and understand more about what's going into our measurements and what our signal actually means, we can answer more questions about diet in the past and human behavior broadly in general. So thank you to my funding sources and collaborators.
The mouse is working, right?
Yes.
Good. No. Sorry. Okay, we set up an experiment about the interaction of magma and water. This can happen during volcanic eruptions, and it may become explosive. The setup is the first that enables us to observe this process on a scale of meters or several meters, which is basically what is the news about the story. There's a little bit more that I will show later. Why do we do this? When magma rises through Earth's crust, it may come into contact with ground or surface water, and this contact may or may not become explosive. One example where it became explosive is the 2010 Eyjafjallajökull eruption in Iceland that disrupted the European air traffic. Another example is the ocean entry of a lava flow that was earlier this year from erupting Kilauea volcano. Over a long period of time, this was relatively mild, but eventually the situation escalated and kind of, no, it's loading, kind of had, well, a pretty explosive event occurred that also affected some spectators that were too close on a boat, and nobody died, but yeah, it's kind of unpredictable. Oh, I just have to stop it. Okay, so the overall goal of our experiment is to understand the causes of this unpredictability and the worst case scenarios of a magma-water interaction. When and why does it occur? What are the chances of these? I'm going to run you real quick through one experiment to get a feeling what this is like. The experiment takes place outside, except from the furnace itself that is housed in to protect it from the weather. About 55 kilograms of material are put into that furnace and are then heated until it is fully molten. Target temperature is around 1300 to 1350 degrees Celsius. The furnace is mounted on a tilting stand. That's the blue legs there. And once the material is fully melted, we pour that material into an insulated container. This is about 60 centimeters high. And that container is then moved away from the furnace to protect the furnace. The container is locked in a position where it's then waiting to be mixed with water. And once it is in that position, all personnel leaves the site and watches from some distance. Yeah, this looks then roughly like that. Then a computer program starts a countdown about 45 seconds. After that, the computer sends out all the signals that are necessary to start the injection system. So move in water injection tubes, start the water flow, release a trigger, which is a hammer that we will see later. And after that is done, after the interaction is done, it will move out the tubes again. I have a little video of that. Is that running now? Okay. So this is, I don't know if you saw that injection system came in from the right side. It's at the lower border. We can watch that maybe later again. Okay, we control the experiment by controlling the amount of water that goes in there, the speed at which we inject it, the positions where we inject it, and the delay of the trigger event. We observe the process with a set of about 5 cameras. That changes a little bit. And from the recorded video material, we compute an intensity of this interaction. And we also collect the ejected material, the products of this interaction, and analyze that, because we can compare these products to natural occurring products to see how they compare. Okay. Yeah, there are some initial results. These results are really initial. I expect the process to become more intense once we kind of optimize it. Initial results include that there is no real trigger event necessary to have an intense interaction. And the intensity of that process depends on the amount of melt on the height of the melt column above the water melt mixing location. Also, we can confirm a minimum mixing speed that makes it very effective. This interaction increases intensity of the interaction at 3 to 4 meters per second. It tends to become very strong. Okay, and I have a couple of more videos because I was told they are cool, so I'm going to show them. I don't see the pointer really well.
You're on it. There you go.
So we have, these are about 20 kilograms, this initial blob of melt. After that, the interaction is somewhat lower, but yeah, we see some mix of water and steam spurting out repeatedly. One question that we try to solve is this hammer thing. It is supposed to start this interaction process or the really intense interaction process, but what we found out is that it may not really be necessary. Sometimes it's still very effective, so yeah. We need to have more of these runs to improve the repeatability. Okay. There's another. This is an overhead camera. It regularly gets drenched. Yeah. Okay. That's it.
Okay, with that, we'll open it up for questions. Any questions from reporters in the room?
I have a couple questions, but I'll just take my turn when I get it. Ryan Williams, National Geographic. To the ancient ecosystem in Antarctica, what kind of speculations sort of exist right now about how plant species survived in such a sunless winter with no sunlight?
I don't know how this works. Is it working now? It's working now. Okay, it didn't get cold enough down there, so everything probably went dormant. The animals have burrows, so they probably hibernated during the dark season, and the plants were deciduous, so they pretty much dropped their leaves and then just went dormant during the dark period. But it didn't really get cold enough. We see no evidence of freezing to show that it was cold enough to cause the dormancy we see today.
And then, if no one else has any questions, the rotting meat. So with all the different nitrogen isotopes that you get from Neanderthal fossils, do you see any geographic variability in the nitrogen from one places versus the other to give you a clue of how different Neanderthals in different geographic locations might have different diets?
So this is actually a really interesting aspect of this. Despite all of the different places that Neanderthals lived and all of the different habitats and different latitudes, the pattern is the same. Throughout all of the different Neanderthal sites, the isotopes come out really, really high. You can only compare nitrogen isotopes within one site, so basically we create a relative scale of where they fall on herbivore to carnivore, and in every single site, they're higher than hyenas, they're higher than cave lions, they're higher than cave bears. And it's consistent whether you're looking at the Near East and Israel, whether you're looking at northern Germany or France or Gibraltar or the UK, it's the same.
So you think that the diets are relatively consistent across?
I think at this point that
at least based on this.
the food behaviors are what is driving this pattern. So it could be what foods are they selecting? Are they all behaving similarly by scavenging part of their diet? Are they all cooking something in a way that increases the nitrogen content? Are they all eating specific types of plants in a certain way? So I think that this is behaviorally driven. This is not a biological or physiological manifestation. This is about behavior, which is what we're trying to get at by doing these experiments.
Okay, do we have any other questions from reporters in the room? Do we have any questions on the chat? Oh, is there one? Oh, yeah.
Hi, Bethany Brookshire from Science News for Students. My question is for Ingo. I was wondering what your awesome experiments showed about the potential predictability of magma explosions upon exposure.
I think they show, at the current stage, they show that they are kind of not predictable, not easily predictable. We cannot just assume that, oh, water is available, so it's going to explode. On the other hand, it may potentially become very explosive, and so there's a large amount of uncertainty.
Do we have any more questions from reporters in the room? Okay, I think that's it. This will conclude our press conference for today, and we'll start again tomorrow morning at 8. Thank you all for coming.